Hello, this is Dr. Deborah Morris, and this is our first lecture in medical physiology. We're going to talk today a little bit about cellular physiology and establish some extremely important baseline knowledge that will be critical during all of this course. As we work through our course in medical physiology, we're going to be talking primarily about human organ systems and how they work when they're working correctly. And um, today's lecture establishes some basic understanding of how all cells work, and how water and electrolytes are distributed within the body, because you'll find that as we talk about systems like the neuromuscular system or the renal system, the cardiac system, understanding these basic concepts is going to help you. It will also help in pharmacology, and one of the things that I've tried to do with course is align my lectures in such a way that we get to certain important concepts in physiology before you encounter them in pharmacology. You're going to be getting a lot of information very quickly this semester, but you'll find that it kind of layers in ways that make it easier to understand. So today we're going to talk about the distribution of water in the organism in something that we will call compartments of body fluid. We're going to understand why there are differences in the concentration of assorted electrolytes in the extracellular fluid compartment and the intracellular fluid compartment. And we're going to talk a good deal about how those differences happen. We're in doing that, we're going to learn about how various substances, including those electrolytes, move across cell walls. The distribution of different electrical charges establishes something called an electric potential difference across cell membranes from the inside to the outside of the cell. And that allows the generation and propagation of electrical signals that we call action potentials that are crucial in the function of nerves and the entire nervous system and muscles as well. We're going to briefly talk about synapses and neurotransmitters and how they create the cell to cell communication that allows these action potentials to spread from one cell to another. And then we're going to talk about how action potentials are coupled to muscle cell contraction. So let's start by talking about water and also electrolytes. As you all know, water is comprises most of an organism, of a human being. And it is the primary component of the fluid that's outside of the cells, the extracellular fluid. And it's most of the fluid within the cells, or the intracellular fluid. And we abbreviate extracellular fluid as ECF and intracellular fluid as ICF. So water is a solvent. And all of the biochemistry that occurs in the organism takes place within this solvent that makes up about 2/3 of the weight of a person, 60%, 65%. Water is most of what we are. And it also acts as a medium through which gases can diffuse, so oxygen on its way in and carbon dioxide on its way out. And for nutrients, it's a solvent through which nutrients like glucose and amino acids can diffuse. And it is the solvent through which waste products like urea and again carbon dioxide diffuse. So, in order for normal physiological processes to happen and to maintain life, we have to have a normal volume of fluid, um, and we have to have a normal composition of the fluids within these two compartments, the intracellular ICF and the extracellular ECF fluid compartments. And this is just a diagram showing you in an adult male who is not obese, water makes up about 60% of the body and solids make up about 40%. If you look at the 60% that is water, so let's say roughly um, 2/3. The intracellular fluid is a larger compartment than the extracellular fluid. And the intracellular fluid is all the fluid that exists within cells. But the extracellular fluid compartment is divided into fluid that we call interstitial, that is between the cells, and fluid that we would probably call intravascular, here defined as plasma and other body fluids, including lymph. So we have the big organism, about 2/3 is water. Of the water, somewhat more than half, and I will call it 2/3, is within the cells. About a third is outside of the cells. Of the fluid that is outside of the cells, about 2/3 is interstitial, and about a third is intravascular. So we have something that we call the rule of thirds, starting with that about 2/3 of the body weight is water. Now, that varies on the body composition, and so the percentage of water is somewhat higher in kids, somewhat lower in the elderly and the obese because there's more lipids in the obese. So here again, about 2/3 of total body water is found within the cells, and about a third is outside of the cells. So 2/3 ICF, 1/3 ECF. Of the ECF, about 2/3 is between the cells, and about 1/3 is primarily plasma, but also lymph, so intravascular. And this is rough, um, but just a way to help you kind of conceive of what happens when fluid volume changes, and we'll talk about that a little bit more as we go on. So, to review, water is more or less 2/3 of body weight. If I were to give you a problem, a calculation, you can round to 60%, just to make it easy, because you can then divide that 60% into thirds, and 2/3 of it is intracellular, and a third of it is extracellular. And of the extracellular, 2/3 is interstitial, and I apologize for missing that. I'll correct it in the PowerPoint slide. And a third is intravascular. And we each of these will consider to be a compartment. And same thing again, so I don't have to go through that again. This diagram just kind of helps to illustrate these subdivisions. So if we consider these cells, the fluid that is within the cells is the intracellular fluid. There is fluid between and around the cells, and we call that interstitial fluid. There is fluid within the blood vessels that we'll call extracellular fluid. Um, so in the extracellular fluid compartment, we have these two additional compartments: the interstitial fluid, which is the fluid around and between cells in all of the tissues of the body, and then the fluid that is within vessels, primarily plasma. But there are some minor subdivisions of what we would of this fluid that is extracellular and not interstitial. So there's, for instance, lymph and lymph vessels. There are fluids in places like the semicircular canals in the ear and the labyrinth there, sorry, the cochlea, that are called paralymph and endolymph. There's fluid around the brain and spinal cord that is CSF, cerebrospinal fluid. There's fluid in our joints called synovial fluid, and we're getting to very small amounts of fluid. There's a little bit of fluid in the space between our the outside of our lungs and the inside of our chest cavity, the between the heart and the pericardium, and in the peritoneal cavity, and then of course there's fluid in the eye. Um, there is exchange of fluid between these various subcompartments of the extracellular fluid that occurs primarily across the capillary lining, which is made up of tissue called endothelium. So fluid can move from interstitial spaces across capillary endothelium into plasma. It can move from interstitial spaces into lymph vessels across the endothelium of lymph vessels, and then from the lymphatic vessels drains directly into the venous system and mixes with plasma. And again, just a diagram illustrating these various compartments. So you have your cells with intracellular fluid. Between them is extracellular fluid that is interstitial. That interstitial fluid um, is part of the extracellular compartment that exchanges continuously across endothelial linings with capillaries and lymph vessels, so into plasma or into lymph, which then enters the plasma from large lymph vessels into veins, into the vein, into the vena cava. So that's sort of my first big concept: fluid compartments and the rule of thirds. I want to move on to another huge concept, which is the idea of homeostasis. This is an extremely critical concept in physiology, and it applies in a number of the in fact, virtually all of the areas that we're going to be talking about as we move through our study of medical physiology, of human physiology. Understand that the body has a large number of mechanisms that help it to maintain a steady state. Okay? In order to stay alive, we have to keep certain things quite stable. And one of those things is electrolyte concentrations. So, electrolytes, sodium and potassium and chloride and bicarb and other circulating ions, their concentrations are tightly controlled. Calcium, for instance, all of these things tightly controlled. And when there is a failure of the homeostatic mechanisms that control those concentrations, cells die and people die. The organism dies. So, in order to maintain homeostasis, there are what we call feedback mechanisms or feedback loops, and most of them are negative feedback loops. So, if the concentration of sodium ions in the extracellular compartment increases. And it's important to understand that all of these mechanisms work based on receptors in the extracellular compartment, not on the intracellular compartment. But if the concentration of sodium ions in the extracellular fluid compartment increases, so sodium levels in the plasma go up, it triggers assorted receptors. And those receptors cause events that will reduce the concentration of sodium ions. Those events can happen in the endocrine system, hormones are released. They can happen in the kidneys, where assorted channels and pumps that regulate sodium concentration of the urine are changed. And similarly, if in the extracellular fluid compartment it is detected that there's an increase in hydrogen ions, in other words, if the pH decreases, there are mechanisms that are triggered that are some of them hormonal, some of them having to do with pumps and channels that we'll talk about later in this lecture, to remove the excess hydrogen ions in assorted ways. And part of a lot of what we'll be talking about in this course is homeostasis and these specific mechanisms. So this is just a diagram showing you a feedback loop. We have a change that occurs in the environment. So that change might be, for instance, an increase in sodium ions in the extracellular fluid compartment in the plasma. There are receptors, and those receptors are in assorted places. Some of them are in the hypothalamus, so the nervous system is very involved here. Some of them are in other places. And they basically detect whether there's a change from normal, from what we would call the set point. That triggers effectors, many of which are glands, some of which are muscles, some of which are in the nucleus of the cell, triggering changes in production of proteins that cause a response to change that internal environment back towards the set point. Okay, so it's a loop, and this is specifically a negative feedback loop. Now, I try not to ask you to memorize a whole lot, and specifically, I'm not going to have you memorize many formulas, even though they can be very important in the study of physiology. But this is medical physiology, and there are some things that you need to know and to remember. Um, this slide has some of those things on it, and I'm going to go through them and talk about them a little bit. But I want you to look here. The important thing about this slide is that the concentrations of these substances are in many cases very different in the extracellular fluid than in the intracellular fluid. So, for instance, in the extracellular fluid, the concentration of sodium is high, and in the intracellular fluid, it's low. And the opposite is true for potassium ions. There's not much potassium in the extracellular fluid, and there's lots of potassium in the intracellular fluid. Even more dramatic is calcium. So while calcium levels are nowhere near as high as as sodium levels, there is a huge gradient between extracellular calcium and intracellular calcium. There's almost no calcium in the intracellular fluid. Okay, so if you were, and these, we will come back to these, but you're going to have to remember that in the extracellular fluid, a normal sodium is around 140 or 145, that a normal potassium is around 4. And those are the only two I'm going to ask you to think about right now. But what's more important is to understand that the concentration of sodium outside the cell is high and inside the cell is low, and that the concentration of potassium outside the cell is low and inside the cell is high. Now, why is that? Why is that? And that's important. And we're going to talk about that. So these are among the important chemical gradients. Intracellular fluid, lots of potassium, not much sodium, quite a bit of protein. There's a lot of protein activity going on inside the cell. In the extracellular fluid, high sodium, low potassium, and glucose. Now we'll come back to how those gradients are established. But first, I want to talk a little bit about the idea of osmolality or osmotic concentration, and how that relates within these fluid compartments. So you remember from chemistry osmolality and osmolarity, and they are a little different. And I have a slide later describing how they're different. But I'm going to say that for the purposes of medical physiology, they are not different, because at the concentrations that we're talking about, these will be essentially the same, and we measure osmolality. If I say osmolarity, I mean osmolality. Okay, so we refer to osmolality in medicine because we can measure osmolality in the clinical laboratory. So the next value that I want you to remember is that osmolarity, osmolality in both the intracellular fluid and the extracellular fluid is about 300 milliosmoles per liter. Okay, and it's the same. It has to be the same. And the reason it has to be the same is that water moves from areas of high water concentration or low osmolality to areas of low water concentration or high osmolality. And so if there is a difference between the osmotic concentration of intracellular and extracellular fluid, which there may be momentarily, relatively transiently under certain circumstances, there is going to be a fairly rapid correction of that because of osmosis of water. So osmosis will eliminate those differences in concentration of water when they occur between the ICF and the ECF. And osmolality under normal circumstances is going to be about 300 milliosmoles per liter. Important value to remember. Plasma membranes are permeable to water. Now, there are channels in most plasma membranes that we call aquaporins that water moves across. And there are some times where that may be less true, and we'll talk about those times because of certain mechanisms in the kidneys, for instance, that help us um, to regulate, to maintain total organism homeostasis. But at this cellular level, water is going to be exchanged um, from areas of high water concentration to lower water concentration across cell membranes by the processes of osmosis and diffusion, um, often through aquaporins or water transport channels. So, one thing that I want you to remember is that all of these mechanisms, all of these homeostatic mechanisms that we're going to talk about in this course, that monitor and adjust anything, but particularly in what we're talking about now, is the concentration of body fluids, the osmolality, the concentration of electrolytes. They respond to changes in the extracellular fluid because the receptors only have access to the extracellular fluid. What's going on inside the cell is not accessible. We can't measure it in the clinical laboratory. Our hypothalamus doesn't see it. Other receptors in vessels in the kidneys and the glands can't see it. So there are no receptors that directly that there are no receptors that monitor intracellular fluid. There are no receptors that directly monitor general fluid balance. Next, cells can't move water by active transport. Water movement is always passive, and it is diffusion and osmosis. And then next, if you increase intake of water or of electrolytes, say sodium, then there will be changes and the body's water and electrolyte contents will rise. Okay, so if dietary intake exceeds losses, you'll get increases. And if losses exceed intake, you'll get decreases. But there are homeostatic mechanisms that will kick in to try to equilibrate. So, here we have a couple of examples. We have our intracellular fluid and our extracellular fluid, about 2/3 and 1/3. They're in balance and they have the same concentration of electrolyte particles. Okay, they're different compositions, right? Because in the intracellular fluid, there's lots of potassium and a little sodium. And in the extracellular fluid, there's lots of sodium and little potassium. But they're in balance. And the solutions are isotonic. Now, suppose you lose water from the extracellular compartment. One way you could lose water is through diarrhea. And our volume, if we lose water in excess of electrolytes, then our volume is decreased, but the extracellular compartment now is more concentrated. There is, relative to the amount of water, there's more electrolyte. And therefore, the concentration of water here is less than the concentration of water here. And what's going to happen is that fluid is going to move from the intracellular compartment across cell membranes into the interstitial and intravascular compartments, decreasing intracellular volume and increasing extracellular volume, and restoring the balance between these two isotonic solutions, so restoring osmotic balance. Um, then here we have an increase in water, I guess. No, this is just showing what's going on under normal circumstances. We have our cells. Within the cells is the intracellular fluid compartment. The cells are surrounded by cell membranes or plasma membranes. Water exchanges across the plasma membranes with the extracellular fluid. There is water that is coming in all the time, being absorbed across the digestive epithelium. So here we have 2 liters of water going in a day, which is pretty normal. As you know, we lose water, and we lose water in our urine, and we lose water through water vapor and evaporation, and we lose water in feces, and we lose a little water through sweat. So if we take in less water and we reduce our extracellular volume and we maintain our amount of electrolyte, it becomes more concentrated. And again, you will have movement of water out of the cells. And that's what this is demonstrating. Here we are bringing in 2 liters of water and we're losing 2,300 mLs of water, more or less. And so some water is going to have to move out of the intracellular fluid compartment into the extracellular compartment. So how does that happen? Well, the plasma membrane is lipids. It's a lipid bilayer, and it has a hydrophilic component and a hydrophobic component. So the lipid is on the inside, and the outside interacts with the water in the extra cellular and intracellular compartments. And we have these substances floating around out here. Ions can't really do much of anything unless there is help. Glucose needs help because it's a pretty big molecule. Water diffuses not very quickly through the lipid membrane, so again, it needs a little help. There are some things that don't need much help. Gases tend to diffuse pretty easily from the water compartment through the lipid membrane back into water. And halothane is a gas that is a anesthetic agent, but we also have obviously lots of nitrogen not doing much, lots of oxygen, a little bit of carbon dioxide that are important metabolically. So ions get rejected, glucose a little bit less so, water diffuses, and these gases make it right through. So how do those things on this side get across the membrane? Let's talk about first diffusion. The gases simply diffuse. Isoflurane, like halothane, is another gaseous anesthetic, but gases can diffuse across cell membranes. These other substances need help, and we're going to talk about the kinds of help that they need. There are a lot of proteins that are studied in this bilayer lipid membrane. Some of the proteins we call channels, and some of the proteins we call carriers. Okay? Channels include aquaporins, which are water channels that allow water to diffuse from an area of higher concentration to an area of lower concentration. In other words, they allow osmosis. There are channels that are specific for ions, and those channels may either allow continuous free passage of an ion, or they may be controlled or gated, and we'll talk about how they may be gated in a few minutes. There are also proteins that we call carrier proteins that are mechanical, that bind a substance and then move them from outside to inside or inside to outside. And finally, and extremely crucially, there are pumps, and these are proteins that use energy to move substances across cell membranes. So here we have an ion channel. This is a gated channel, and it can either block the movement of potassium or, when triggered, allow the movement of potassium. Carrier proteins bind the substance and then change conformation to move it. Now, things that move through those channels result in simple diffusion, and these are ungated channels. Things that move directly across the membrane by dissolving in it, like gases, that simple diffusion. There is diffusion that is called facilitated diffusion with certain carrier proteins that don't use energy but simply bind and change conformation when bound to move a substance across the cell membrane. So there's simple diffusion and facilitated diffusion. But then there's something that we call active transport. And there are a couple kinds of active transport that use energy being ATP, right? ATP, adenosine triphosphate, is a molecule that is produced through metabolism and is used in lots of ways by the cell. And one of the ways it's used is to actively transport substances. So diffusion occurs down a concentration gradient. If you have a lot of oxygen out here and not much oxygen out here, and it's going to diffuse or move from an area of high concentration to an area of low concentration. If something's happened that your, say, extracellular fluid compartment is extra dilute because you drank 2 gallons of water on a Facebook challenge, and so there's a lot of water out here and less water in here, then you're going to get water moving down the concentration gradient from, there's a lot of water out here and less water in here, and you're going to get water moving down the concentration gradient from its higher concentration to its lower concentration. And with facilitated diffusion, same thing. There's a lot of glucose out here. There's not much glucose in here. You will get glucose moving. And that's why we're showing double arrows here for the two kinds of diffusion. I guess it's 3 kinds. Simple diffusion across the membrane, diffusion through a channel protein, and facilitated diffusion. But then when we're talking about active transport requiring specific carrier proteins or pumps, It occurs against a concentration gradient, and it's always going to be in the same direction, from the area of a lower concentration to an area of a higher concentration against the concentration gradient. So, simple diffusion includes lipid soluble molecules that readily cross the membrane depending on their lipid solubility. In addition to these gases, alcohols have high lipid solubility and move easily across cell membranes, don't need any carrier or channel. But water and water soluble molecules that are hydrophilic, they cross through channels or pores, and larger molecules tend to move more slowly. So here we have our various things moving across those channels. Now, channels across which ions move can either be ungated or they can be gated. Okay? And an ungated channel allows the substance to cross, determined by size and shape and the distribution of charges. So obviously ions are charged, and distribution of charges is going to affect the crossing. And we'll talk about that a little bit more later. But gated channels are gated either by voltage or chemically. So there are voltage dependent sodium channels that only open when there's a change in the voltage across the cell membrane. And here we're showing a voltage gated sodium channel that opens when the voltage changes, allowing sodium to cross. There are also chemically gated channels. Many of these are triggered by things like neurotransmitters. So there are acetylcholine receptors, nicotinic acetylcholine receptors, and we'll talk a lot more about these later. Receptors that are channels that allow, when the acetylcholine comes in, to the movement of ions across the channel. Sodium or potassium, different ions. Okay. Now, there is a kind of diffusion that uses proteins studied in the cell membrane that we call facilitated diffusion. I talked about how sometimes when a molecule binds to a protein, the protein changes its shape, allowing the molecule to be moved to diffuse from an area of high concentration to an area of low concentration. And this doesn't use energy. Obviously, energy was previously expended in order to make this protein, but once this protein is there, if there is more of a substance and there are a number of transporters for glucose and amino acids, and also for chloride, that will move those substances from an area of high concentration to an area of low concentration through this mechanism called facilitated diffusion across the membrane through a channel that changes conformation once it binds the molecule, and that goes, because it's diffusion, from high concentration to low concentration and does not use energy. Simple diffusion depends on the size of the molecule, but mainly on the concentration gradient. So the higher the concentration gradient, the higher the rate of diffusion. Okay, but facilitated diffusion is dependent on how many receptors there are, how big the molecule is, and how long it takes for the substance to bind to the receptor, to the channel, and then how long it takes for the shape change, the conformation change, in order for the substance to be diffused and released on the other side of the membrane. So simple diffusion, entirely dependent on the concentration gradient. Facilitated diffusion, dependent on the size of the molecule, the number of the receptors, and the time for binding and shape change. So let's talk about those factors that affect the rate of diffusion. This is simple diffusion, showing a downhill movement across a pore from an area of high concentration to an area of low concentration. And how quickly it happens just depends on the concentration gradient. As the concentration gradient is relieved as it approaches similar concentrations, the rate of diffusion is going to slow. But electrical potential can also affect that. Okay, so here we have a bunch of charges. Okay, we have a battery. We have, I guess, two terminals, positive terminal and a negative terminal. And right now we have within this area positive charge and outside a negative charge. Okay, so because there's a positive charge inside the cell, you're going to have negative charged particles, let's say chloride, moving down the electrical gradient from towards the positive charge. When will the negatively charged molecules stop entering the cell? Well, it happens when this electrical gradient, which we kind of can think of as a force, equals the force of the chemical gradient. And that's called the equilibrium potential or the Nernst potential. So the chemical gradient and the electrical gradient end up being in equilibrium. It's not continuous. You have to have, you reach equilibrium between the electrical gradient and the chemical gradient. And we'll come back to that in a little while. A third thing is the pressure difference. If there's a higher pressure, then there's an increased energy to cause net movement from a high pressure area to a low pressure area. And an example of this would be in the kidney, where in the glomerulus, the pressure in the capillary is higher than the pressure in the glomerular space, and therefore fluid and electrolytes, things that easily move across the cell membrane, get pushed, and there's net movement from the high pressure to the low pressure area. The reason that even electrolytes are moving there is that those cell membranes in the capillaries in the glomerulus are kind of holy, so small molecules move easily. And we'll talk a lot about that when we get to our renal section. Water moves by osmosis. So osmosis describes the diffusion of water. And it's always going to move from the higher water concentration to the lower water concentration. So what does that mean? Here we have a membrane with pores in it, and we have pure water on one side and a salt solution on the other side. If you look, the salt in this solution means that there's a lower concentration of water. And therefore, water molecules are going to move from the area of high water concentration across this semi-permeable membrane. And it's semi-permeable because it's not letting the sodium and chloride ions move back. So water is moving across this semi-permeable membrane from an area of high concentration to an area of low concentration. People get confused about this, and I want you to remember it's the water concentration, not the concentration of solute. And water is always going to move from an area where it's purer to an area where it's less pure, from the high concentration of water to the lower concentration of water. And that creates conceptually a pressure that is attributed to the osmolarity of a solution. And it's not the size of the particles, it's the number of the particles. So if you have water and salt solution, the water is going to move across the membrane, the semi-permeable membrane, into the salt solution, and you're going to end up, it's going to continue moving until the physical pressure of this column of water equals the osmotic pressure created by the difference between the pure water and the salt solution. So water is going to move until the pressure in this column equals the osmotic pressure. And this is the slide I mentioned, that osmolarity means the osmoles, particles per liter of a solution, and osmolality is the osmoles per kilogram of solute. And so they're very similar. And at the concentrations that we're talking about, they're essentially the same. So we can measure osmolality, and that's the term we use medically, is osmolality. And again, in normal body fluid, both intracellular and extracellular, osmolarity is about 300 milliosmoles per liter. And we can only measure that, and receptors in the body can only detect that in the extracellular solution. So let's do a quick case. Our patient is a long-time smoker who's 64 and was recently diagnosed with small cell lung cancer. And he comes into the ER after having had a seizure at home. And he's confused, and we draw labs. And his sodium is 124. Now, if you remember, I said that normal sodium is, say, 140 or 145. And along with his decreased sodium, his serum osmolality is decreased. Let's say it's 270 or 260 instead of 300. Now, I want you to think about why he's confused. So this is pretty complicated. Why is he confused? Well, confusion has to do with the central nervous system, right? With his brain. And if his serum osmolality is low, It means there's less solute, less sodium in his extracellular fluid. And since we start with equilibrium, the intracellular fluid was at 300, but water is going to be moving from an area of higher concentration, or the extracellular fluid, to an area of lower concentration, or the intracellular fluid, which means his brain is going to swell. And his seizure and his confusion are currently due to increased intracranial pressure. And we need to do something about that. So, because his sodium is low, we give him some saline that is stronger. It's hypertonic. It's more concentrated than the normal saline that we give. What we call normal saline is 0.9% saline solution, which is about the same osmolality as serum. It's about 300. And so it doesn't, when you give normal saline, it does not change serum osmolality. But he needs his serum osmolality increased. So we're going to give him a saline solution that's has higher osmoles of salt, right? And we give it through a central line into a big vein. And my question is, do you think we need to do this quickly or slowly? And why? So think about that for a minute. And again, just sort of thinking about homeostasis, this is something you want to do slowly. You do not want rapid shifts of water between the intracellular and extracellular fluid compartments, particularly in the central nervous system, because you could overshoot. So we don't want to shrink the brain quickly. We want to slowly raise the osmolality of the extracellular fluid to allow water to move back out of the cells, slowly decreasing the swelling in the brain that caused the seizures and the confusion. Now, you may be wondering why this is, and I'm going to tell you that this is something called the syndrome of inappropriate ADH. ADH is a hormone, antidiuretic hormone. Antidiuretic hormone keeps you from losing water. It makes you not have aquaporins in your kidneys. And so water stays in the body in excess of sodium, and you get a high water concentration and a low sodium level. And there are some lung tumors and other tumors that make substances that essentially act just like ADH and therefore cause this SIADH, syndrome of inappropriate ADH. Another thing that can cause SIADH is a head injury that makes your brain make more ADH accidentally, causing again an increase in water retention, a decrease in serum plasma osmolality, causing central nervous system issues. Okay, so let's get back into leave our little clinical break and get back into our lecture. We talked about diffusion. Now we're going to talk about active transport. Again, active transport uses proteins that are in the cell membrane. But in opposition to the idea of facilitated diffusion that doesn't use energy, or simple diffusion that doesn't use energy, active transport uses energy. And there are two kinds of active transport. The first is called primary active transport, and it directly uses ATP to pump molecules from an area of low concentration to an area of higher concentration, so against the concentration gradient. And that directly uses energy. It costs energy, energy being ATP. And an example of this that we'll talk about that is probably the most important example is the sodium and potassium ATPase, or sodium potassium pump, that moves sodium out of the cell and moves potassium into the cell using ATP. That's primary active transport. There is also secondary active transport, which uses the concentration gradient of another molecule, usually sodium, that was created by the direct use of energy. So the concentration gradient of sodium was created through primary active transport. But now that we have that concentration gradient, we can use the energy stored in that concentration gradient indirectly to move substances from an area of lower concentration to an area of higher concentration, and that is called secondary active transport. And it indirectly uses energy. It uses the energy stored in the concentration gradient of sodium. So let's talk first about the most important pump. And that pump is what we call the sodium potassium ATPase. And it has a really important function, which is that it creates this concentration gradient of potassium and sodium across the cell membrane of every cell in the body. Okay? And that allows us to maintain our normal osmotic balance by maintaining this balance between sodium and potassium. It uses between 1 and 2/3 of the energy of, again, every cell in the body. Some cells it's more crucial and uses more energy. But understanding it is important for understanding how a ton of things work. It's extremely important in the kidneys, and it helps us understand how diuretics work. It's extremely important in the heart, and it helps us understand how certain heart medicines, particularly the digitalis compounds, work. And it's extremely important in the neuromuscular system, and helps us understand action potentials, how we create the movement of electrical signals along nerves and muscles, causing muscle contraction. So this pump, this sodium potassium ATPase, it can be called a pump, it can be called a carrier, it can be called a transporter. It allows, by the use of ATP, and it's called an ATPase because it's an enzyme that breaks ATP into ADP, right? So this pump or transporter or carrier will move 3 sodium ions out of the cell and 2 potassium ions into the cell, using an ATP molecule that is then broken down into an ADP plus a phosphate. Okay, so that creates the gradient of high potassium inside the cell and high sodium outside the cell. Low sodium inside the cell and low potassium outside the cell. But one of the things that I really need you to remember, this is if I put something in red, you can kind of expect it to turn up in a test question. But that 3 sodium ions are exchanged for 2 potassium ions. And I want you to think about that a little bit because chemically, sure, that works. But doesn't that do something electrically? We're moving 3 positive charges out and 2 positive charges in. So we'll come back to that in a little while. Other pumps or transporters include the calcium ATPase, which is super important. Remember I mentioned that big gradient between calcium high outside and very, very low inside the cell. It's present on the cell membrane, but also on the membrane of the sarcoplasmic reticulum in muscle cells and on mitochondria, so that the calcium concentration in the cell solution, the cytosol, the intracellular fluid, is very, very low, but it's higher within the sarcoplasmic reticulum. So within the sarcoplasmic reticulum of muscle cells, and we'll talk about why that's important later. There is a hydrogen ion, ATPase, and it's found notably in the parietal cells of the gastric glands, which is how we make a hydrochloric acid solution in the stomach as sort of one of the early steps in digestion. It's also found in cells of the renal tubules that are called intercalated cells that help us to control the pH of blood and through feedback loops, as I mentioned earlier. It can concentrate hydrogen ions enormously, particularly in the gastric secretions. So hydrogen ions are concentrated up to 1,000,000-fold between the hydrogen ions that are on the intracellular side and on the extracellular side in the stomach solution. So let's talk about secondary active transport. We mentioned earlier that there were 2 kinds of active transport. Well, there are 2 kinds of secondary active transport, co-transport and counter-transport. In the case of a co-transporter, which is also called a coporter, the substance moves in the same direction as the ion that's driving it. So I mentioned that most of the time potassium is the ion whose concentration drives secondary active transport. The driver, the sodium, has a gradient because it was actively transported using the sodium potassium ATPase. So there's a high concentration on one side of the membrane, the extracellular side, and a low concentration on the intracellular side. And it allows another substance to move with it in the same direction. So no ATP is used. The movement is secondary to the gradient of sodium that was created by the sodium potassium ATPase. That's a coporter. And here we're showing some coporters. So sodium and an amino acid moving in the same direction, sodium and glucose moving in the same direction, sodium and 2 bicarbs moving in the same direction. HCO3 is the bicarbonate ion. So there are assorted coporters use secondary active transport. I mentioned earlier that there are carriers that facilitate diffusion of amino acids and glucose. There are also secondary active transporters, coporters, that allow the passage of amino acids and glucose using the help of the sodium gradient from the outside to the inside of the cell. I didn't make these cute little animations, but I kind of like them, so I leave them. There is also counter transport. These are called antiporters, in which case the protein that is the porter uses the driver ion to transport a substance in the opposite direction. So again, it's transporting whatever substance it is from an area of lower concentration to an area of higher concentration, but it's using the gradient of sodium, which is moving from an area of higher to an area of lower. And these are examples. There is a calcium sodium countertransport, a sodium hydrogen ion countertransport, and there's a countertransporter that uses the combination of sodium and bicarb to transport chloride and hydrogen ions from the outside to the inside of the cell. Okay. And this is just as a couple of examples. So in the intestine, there is an epithelium. So these are cells that are lining the intestine. And on the lumen side of the intestine, there's something we call the brush border that greatly increases the surface area. And there are substances that are being moved from the lumen of the intestine through the cells of the epithelium and into the intracellular fluid. On the luminal side, okay, there is a constant movement, if you remember, of sodium out and potassium in, and that's on this is on the outside of the cell. So not the lumen side, but on the extracellular fluid side. So on that side, the sodium potassium ATPase is continuously moving 3 cells out, 3 ions of sodium out and 2 ions of potassium in. Okay. On the luminal side, there is secondary active transport that is using the high concentration of sodium, in this case in the lumen, to co-transport both amino acids and glucose into the cell. And then there is facilitated diffusion, moving it from the inside of that cell into the extracellular fluid. So, questions about those transports. That was a lot of material. I'm hoping you're taking this in chunks. I should probably be recording it in chunks. I want to now talk about electrical gradients, and this will come back to some of the things that I kind of pointed out as we were going. As you know, there are a lot of substances that when they're dissolved dissociate into charged particles, into ions. So for instance, sodium chloride, when dissolved in water, becomes positively charged sodium ions and negatively charged chloride ions. And when we move those ions with pumps, we can create electrical gradients along with the concentration gradients. So when we are moving 3 sodium ions out of the cell and 2 potassium ions into the cell, we are getting a electrical gradient because we have one more positive on the outside of the cell than we have on the inside of the cell. So obviously, looking at the big picture, charges always balance. But on the tiny scale of cells and cellular membranes, the movement of these ions can create electrical gradients across cell membranes along with chemical gradients. I talked about a concept called the equilibrium potential. And the equilibrium potentials for potassium and for sodium are extremely important in cellular function in a lot of ways. So let's look at what happens. We have the sodium potassium ATPase that is establishing a chemical gradient, okay, where we have on the outside of the cell low concentrations of potassium and on the inside of the cell high concentrations of potassium. We also have some ion channels that are specific to potassium because there is a lot of potassium in here and not much potassium out here. You're going to get a movement of potassium ions down its concentration gradient through those channels, removing even more positive charges from the inside of the cell. So we now have this gradient where the outside of the cell is positively charged because of the sodium potassium ATPase and also because of potassium channels moving potassium ions down their concentration gradients. So we have a positive charge on the outside of the cell and a negative charge on the inside of the cell. But just as there is a kind of pressure from the concentration gradient, there is also this, because of this membrane potential and the negative charge on the inside of the cell, those potassium channels have two-way movement. You're going to have some potassium flowing back in from this positively charged outside to the negatively charged inside. And it's when those the chemical and the electrical gradients balance that you reach something called the equilibrium potential for potassium. So let's review that. The sodium potassium ATPase creates an electrical gradient by itself because it moves 3 sodiums out for every 2 potassiums that it moves in. So you have a slight negative charge inside the cell. There are also channels across which potassium moves, and that increases that electrical gradient because more potassium moves out of the cell down its concentration gradient than moves into the cell. And it's when the electrical potential and chemical potential or gradients balance that you reach this thing called equilibrium, or the equilibrium potential for this single ion potassium. And we know it's not the only important ion, and we'll come back to that in a minute. So the negative charge inside the cell is pulling potassium in, and the concentration gradient is pushing potassium out. Okay? And when they balance, we have something called the equilibrium potential. And that is in many cells about negative minus 90 millivolts. So everything's kind of working to balance. First, the sodium potassium ATPase, and then the potassium channels through which potassium is moving in both directions. And when the concentration gradient and the electrical gradient balance, you've reached the equilibrium potential, which is approximately minus 90 millivolts. Okay. There's also an equilibrium potential for sodium. So obviously the potential is established by the sodium potassium ATPase pumping out more positive charges than it pumps in. There's not a lot of sodium channels. They're not open all the time like the potassium channels. But there's a little bit of movement of sodium down its concentration potential. And the two equilibrium potentials for sodium and potassium kind of operate together. There are fewer sodium channels than potassium channels, and so you end up with something called the resting membrane potential. And just you don't have to know all of the mechanisms, but we have the sodium channels, not a lot of them, usually closed. We have the potassium channels, more of them mostly open. There are some chloride channels. We have some anions that are organic on both sides, so we end up with this charged membrane, positive on the outside, negative on the inside, that ends up at its resting membrane potential being in the neighborhood of minus 60 millivolts. And that's measurable. So this is stuff that physiologists did 100 years ago. So why is it important? Well, it's important because some cells use that potential to allow, by changing the membrane potential, to allow the movement of electrical signals and chemical signals that result in the functions of the nervous system and of the muscular system. So without that electrical potential, the resting membrane potential, we wouldn't have a nervous system, we wouldn't have functional muscles. So I want to talk about a few concepts in the changes that can occur in the membrane potential. The establishment of that membrane potential is called polarization. So the cell wall becomes polarized. It is positive on the outside, it is negative on the inside, and the process during which that happens is called polarization, and it achieves resting membrane potential. And here we have the things being pumped causing polarization. You can actually overpolarize, all right? You can overshoot. When there is movement, polarization is just the establishment of the membrane. Depolarization is when suddenly there is movement of that membrane potential back towards zero, and that can overshoot. Repolarization is the reestablishment of the resting membrane potential by the movement of potassium ions back out of the cell. So there are things that happen that cause the movement of sodium ions into the cell that depolarize, and then the re-establishment through the pumping of sodium out and potassium in, um, or sorry, sodium out and potassium in. And there is a period called hyperpolarization when the cell membrane is a little bit more negative than resting membrane potential, but then it returns to resting membrane potential. This happens in small increments of time. Things can happen to cause this depolarization, okay? And the stimulus that changes a membrane potential can be weak or strong. There are channels, we've talked about gated channels, ligand gated channels that open when something binds to them. And it could be a neurotransmitter like acetylcholine. And when that channel opens because of binding of acetylcholine, an ion moves, and here we're going to say sodium, that changes the membrane potential depending upon the amount of the stimulus. So if you have a little acetylcholine, you get a little movement. If you have a lot of acetylcholine, you get a lot, and you get the membrane potential closer to 0 millivolts. But when it reaches a level called threshold, you suddenly get an active potential where voltage gated channels open. And those voltage gated channels open or close depending on membrane potential. So here you're at resting membrane potential, and maybe you're getting some little signals, some little signals from a neurotransmitter. But once they reach threshold, enough channels have opened from the ligand gated channels. Suddenly, the voltage gated channels open. Okay, and you get depolarization. So, when, say, enough of the ligand gated channels have opened to move the resting membrane potential from, in this case it says -90 to -65, that's threshold, and suddenly the voltage gated sodium channels open. Sodium starts rushing into the cell, returning that cell membrane potential to close to 0. A little bit of overshoot. When the sodium channels close, you get potassium channels that are also voltage gated that open. And you also have the continuous action of the sodium potassium ATPase. So now you have a return of the polarization back a little past the resting membrane potential, and then returning to the resting membrane potential. And this moves along, it propagates along the membrane of an axon or of a muscle fiber to allow the transmission of signals. In the resting state, all of the channels are closed. There is ion movement, but it is stable ion movement. There's no permeability. So you have the normal pumps working to maintain the resting membrane potential, and you have a flat curve. During depolarization, once you've reached threshold, the sodium channels open and sodium flows into the cell, and you have a sharp upward spike of the action potential curve. When you reach overshoot, the sodium channels close because they are voltage gated and the voltage is now changed. The potassium channels open, potassium flows out of the cells because it's flowing down its concentration gradient, returning the membrane potential back towards a negative membrane potential, and you have a downward movement of the action potential curve. And then during hyperpolarization, there's a reset. You still have some potassium flowing out, so you get a slight dip below the resting membrane potential. And here we're going to show those steps. So at 1, you're in the resting state. Sodium channels, which are voltage gated, are open. Potassium channels, they're closed. Potassium channels are closed. You reach threshold. The sodium channels open. Sodium rushes into the cell. Okay. And you depolarize. So you have this rapid change. Okay, this is reaching threshold. That's step 2. So this is some sodium channels are open. Probably these are the ligand-gated sodium channels that are responding to something like either small changes, electrical changes, if they're gated by voltage, or binding of neurotransmitters if they're gated by chemical. And once you reach threshold, because the membrane potential has changed, has gotten a little closer to 0, and it's gotten to the threshold potential, then all the sodium channels open. Sodium rushes into the cell. The membrane potential returns to 0 and past 0, and that's your action. That's the depolarization phase of the action potential. Then the sodium channels start to close, okay, and the potassium channels open, and now potassium is leaving the cell, and you are returning, getting rid of positive charges, returning towards and a little bit past the resting potential. You get a little bit of hyperpolarization, and then finally get back to the resting state. So things to know: that an action potential, like you can get small changes in potential, but an action potential is yes or no. It's all or none. It's 0 or 1. Once the threshold potential is reached, you're always going to get an action potential, and it's always the same. It's the same strength. So the graded potentials that help us to reach threshold, they vary, but they don't trigger the action potential until you get to threshold. You can have graded potentials that are additive, increased in frequency, or they can be weaker or stronger, like more or fewer molecules of a chemical ligand. But once it reaches the threshold potential, it triggers an action potential. Once an action potential occurs, there's a refractory period while everything resets, during which you can't have another action potential. So here we're showing a stimulus. We're at resting membrane potential. We get a little stimulus, it doesn't do much. We get a little another stimulus, it doesn't do much. We get a stimulus that brings us to threshold, and we get an action potential. And depolarization, hyperpolarization, repolarization, a little bit of undershoot, and then we're back to resting membrane potential. Our next stimulus is big enough to trigger another action potential, and it's going to be exactly the same. Doesn't matter. These small subthreshold stimuli don't affect the action potential. If this particular stimulus was up to here, that it doesn't matter. You wouldn't get a bigger action potential. It's always the same. The action potential is always the same. And the last thing we're going to talk about are neurons, synapses, and the neuromuscular junction. So we'll come back to this much later in the course. But neurons have a cell body with a nucleus in it. They have structures called dendrites, and they have a single structure called an axon. Okay. Between dendrites, or between axons and dendrites, you can have a place where they don't touch, but they come in close proximity, called a synapse. And at the synapse, neurotransmitters can be released, they can bind to receptors, and signals that are transmitted electrically along dendrites or axons then suddenly shift to a chemical signal, which then triggers through chemically gated receptors another electrical signal, which then passes along the dendrite or the axon. Within the presynaptic cell, at the synaptic terminal, there are vesicles that are full of neurotransmitters. Those vesicles, when triggered by an electrical signal, merge with the cell membrane and release their neurotransmitter molecules into the synaptic cleft, where they are picked up by receptor sites on the postsynaptic cell. Those neurotransmitters might be taken back up into the presynaptic cell and restored in vesicles, or they might be deactivated in the cleft. So just briefly, this is how muscles work. The axon terminal of a motor neuron has within it a lot of vesicles, synaptic vesicles, that are full of a neurotransmitter called acetylcholine. When an action potential propagates along this axon, because as the voltage moves along the axon, it is opening new voltage gated channels, allowing propagation of the action potential along the axon. When it gets to the neuromuscular junction, synaptic vesicles merge with the cell membrane, releasing acetylcholine into the synaptic cleft at the neuromuscular junction. So this is the axon part of the neuron, and this is the muscle cell. And within, which is called here, the sarcolemma, is the cell membrane of the muscle cell. When acetylcholine is released into this synaptic cleft and picked up by receptors on the sarcolemma, on the muscle cell membrane, it releases calcium ions. It opens chemically gated channels that cause the release of calcium ions from the sarcoplasmic reticulum and the extracellular fluid into the cell, the muscle cell, allowing contraction of the actin and myosin fibers, and the muscle then contracts. As long as acetylcholine is being released, the muscle will contract. As soon as release of the acetylcholine stops, it is getting continuously inactivated here, and the muscle will relax. So it's inactivated by an enzyme called acetylcholinesterase. So here we have a axon at the motor end plate. So the axon synaptic terminal is releasing acetylcholine, which is binding to receptors. There are things called T-tubules, which allow the extracellular fluid kind of to get into deeper contact with inner parts of the cell. Also, there is the sarcoplasma reticulum. So when the acetylcholine binds to receptors, we open gated channels, allowing the release of calcium, which binds with actin, allowing the actin to interact with the myosin and contract the muscle. At the neuromuscular junction, the acetylcholine is removed by acetylcholinease. The calcium ions are pumped back into the sarcoplasmic reticulum and into the extracellular fluid. Um, we have recovered active site, so the actin is not interacting with the myosin, and contraction ends and relaxation occurs. So, just a brief clinical discussion. We have a patient who's a 43-year-old woman who we know has a disorder called myasthenia gravis, and she's complaining of double vision and difficulty holding her head up. And when you look at her, you see that she has drooping of her eyelid and also some unusual movement of her extraocular motion so that her eyes are pointed in different directions. So when you look at her chart, you see that she has a positive test for anti-acetylcholine receptor antibodies. Why do you think she's having muscle weakness? And this is an autoimmune disorder in which the immune system makes antibodies that attach to acetylcholine receptors, not allowing the acetylcholine to bind and to cause muscle contraction. And as you can see, it doesn't have to be the same at every muscle. And so she, at the moment, is having difficulty with holding her head up. She's having difficulty with vision. And I'm going to stop here. We will come back later to the nervous system structure, the nervous system function, and much later to the musculoskeletal system. But because of what we're going to be doing in terms of neurological function and cardiac function, we needed to have a little bit of understanding of the importance of action potentials, how nerves fire, how muscles contract, and the concept of neurotransmitters. Please feel free to email me questions, and hopefully we'll have lots of time to talk about this.